And here we are. This is official. So I would like to thank you again for joining our thematic international webinar on bioeconomy applications of bio-based products within the Interreg Adrion Bioeco RDI project. As you know, it will be comprised of 3 sessions. Session 1 will be on aquaculture and fisheries waste and byproduct management by myself, Natalija Topić Popović from Ruđer Bošković Institute, Zagreb, Croatia. Hello, Andrea. I would kindly ask you to switch off your camera if possible. Hello, Andrea. Can you hear me? Thank you. The session 2 will be on Nanocrystalline Cellulose production and applications by Matjaž, Kunaver, National Institute of Chemistry, Ljubljana, Slovenia. And session 3 will be on optimal use of waste streams for biogas production by Valantis, Ketikidis, Center for Research and Technology, Hellas, Thessaloniki, Greece. So each session will last for 15 minutes. Should you have questions, please enter them in the public chat area and the presenters will address them by the end of the webinar. Once again, please mute your microphones and switch off your web cameras. The webinar is recorded and you will be able to download it from your homepage. So we go to the first presentation on aquaculture and fisheries waste and byproduct management. I would like to stress that biomass deriving from fisheries and aquaculture are important sources of nutrition and livelihood for millions. In recent past, fish byproducts were considered to be of low value and were only used for animal feed or discarded as waste. The interest in utilization of fish byproducts has been on the rise during the last 20 years because of their potential as a significant additional source of nutrition, as well as a source for other needed products. Fish processing is a very important sector because it adds value to the primary product. But it produces great amounts of byproducts. 25 up to 70% of fish ends up as a byproduct, which are still not utilized in sufficient quantities to increase their economic potential. The European Union is the 4th largest producer in the world, producing a little over 3% of fishery and aquaculture products. Structurally speaking, 80% of this quantity originates from fisheries and 20% from aquaculture. The leading countries regarding catches are Spain, Denmark, the UK, and France. China is by far the largest fishing nation, with production of almost 79 million tons, or 38% of global production. Totally opposite than EU, its catches represent only 23% of Chinese production, but it is still the country with largest volume of catches in the world. It is followed by Indonesia and then by India. As we can see on the image, globally half of the consumable fish is farmed, while half derives from fisheries. In the near future, this ratio will change in favor of aquaculture. Of the total fish production, 88% is utilized for Nicola, please switch off your microphone.
Excuse me.
88% is utilized for direct human consumption, while the remaining 12% is used for other purposes, such as fish meal and fish oil, the vast majority, or 74%, as material for feeding in aquaculture, for farming of livestock and fur animals, for further aquaculture purposes, as bait, in pharmaceutical and ornamental purposes. Fish processing creates byproducts up to 70% of fish, like we said, and byproducts have an enormous potential for development into products. However, fish byproducts are usually not placed on the market due to poor consumer interest or because sanitary regulations restrict their use. Therefore, fish byproducts need upgrading. There are 2 main types of upgrading. First is the mass exploitation. The main sectors deriving from this approach are fertilizers, energy, and feed. And second, the small volume exploitation for higher added value for sectors as nutrition, health food, nutraceutical, cosmetic, or pharmacy. They have different characteristics that can be synthesized by the figure. The main differences between those upgrading ways are the capacity of absorption by the market and the profit that can be achieved. At the top of the pyramid, we find, as it is expected, the sectors corresponding to the small volume exploitation. These activities do not need a lot of volume, but they are more constraining in the quality terms. They require rigorous sorting, low temperatures, and rigorous traceability implementation. However, fish are not the only aquatic animals contributing to its biomass, production, waste, and byproducts. Shellfish are extremely interesting group in that respect. Shellfish waste is mostly generated from production processes, and it amounts to 6 to 8 million metric tons annually. Up to 80% of shellfish can turn into discards. Shellfish wastes are disposed in landfills or thrown back to the sea, processed into feedstock, or used as a fertilizer. Of course, landfilling or sea disposal can cause many environmental issues. As you can see, shellfish waste consists of many extremely valuable compounds, which can be used as antimicrobials, antioxidants, thickening agents, flavor enhancers, emulsifiers, for wound healing, anti-tumor drugs, anticoagulants, heavy metal absorbents, and many, many other purposes. To name just a few, I would emphasize the value of carotenoids as powerful antioxidants and immune enhancers, proteins with essential amino acids, and the nutrient value comparable to soybean. But the most valuable compounds from shellfish are chitin and chitosan, offering a wide variety of biological and industrial applications such as medical and pharmaceutical implants, tissue engineering, etc. So why not use shellfish waste and byproducts, although we have large amounts at disposal with potentials for pharmaceutical purposes and green solutions? Many would argue that the methods for their exploitation are dubious. The industrial processes for shell conversions more often than not use hazardous chemicals, and shellfish waste management is still widely not regulated. Further on, shellfish farmers are required to store and separate generated waste, which should be recovered or disposed of. They are required to hand over their waste to companies for waste management. However, if the annual production on a shellfish farm is below 400 tons and it is not located in a protected natural environment, the farmer is required to handle waste and take care of environmental measures himself, which makes an additional impediment to their entrepreneurial initiatives. And so we return to fish. All fish in the EU which are fished out under quota or at the minimum size need to be landed. They cannot be used for human consumption and are mostly converted into fish meal and fish oil or as feed in tuna farms. Of course, the potential is to use this biomass also in pharmaceutical, cosmetic, and in other purposes. Now, coming to the type of waste from fisheries. You can see that catch under minimum size cannot be used for human consumption, and it is treated as a resource. Also, animals with the disease signs transmittable to humans need to be destroyed or used for products, but it is also allowed to return them to the sea, as well as discarded catch, damaged fish, and byproducts of processing. In the other image, you can see that shellfish affected by diseases cannot be returned to the sea, but it is allowed to use them for products. The category 2 shellfish have dual possibilities: to be destroyed as waste, but also to be used for landfills, fertilizers, biogas, and for other products. In aquaculture, less biomass is allowed to be returned to the sea, and it offers a lot of potential for biotransformation. Although some would argue that discharging of aquatic biomass to the sea has advantages, particularly in returning organic matter to the marine food chain, the disadvantages prevail, as such biomass can endanger the marine environment and it presents an enormous potential for production of valuable products. Therefore, in order to avoid wasting of valuable biomass and its throwing back to the sea, we need to encourage its collection. For that purpose, education of good waste management needs to be conducted, as well as to create logistical preconditions. If needed, offer cash incentives to those who collect and process such biomass, encourage investments in processing, and finally build a waste management center for aquatic biomass. That is something that our partners from Agroproteinka company, which some of you have visited, would also like to build as an addendum to current facilities. And finally, all the data presented and much more in a more structured and elaborated form are presented in the Croatian regional strategy and bioeconomy of byproducts and waste from fisheries, aquaculture, and processing industry, which was a deliverable of this Bioeco RDI project. And with this, I would like to wrap up this session and move to the next one. Matjaž Kunaver from National Institute of Chemistry in Slovenia will tell us a few words on his work with nanocrystalline cellulose. Let me switch the microphone to you, Matjaž, and please carry on in a few seconds. Just a minute. Matjaž. There you go, Matjaž.
Okay, thank you. Hello, everyone from my side as well. It's my turn to talk about nanocellulose. This is the hottest research subject worldwide, except perhaps COVID vaccine, which is now even more popular. I would like to show you one of successful transfers from research to industry, from laboratory work experiments to pilot plant production, and some applications with outstanding results. Okay. The biomass is, of course, an immense and renewable resource all over worldwide, and can be actually a source for biofuels, valuable chemicals, and nanocellulose as well. And this is the topics that I would like to talk about a little bit more in details. You know that much efforts has been devoted to converting these types of biomass into useful industrial and commercially viable products. In recent years, some effective processes have been found, such as thermochemical conversion, producing several new products from these sources, including some basic chemicals. On the other hand, we know that biomass is spreading, and someone made a calculation. 170 billion tons of biomass is produced annually. So it is a really great amount of material that can be converted in some chemicals or some useful stuff. Slovenia is the most forested country in Europe, and the forest is spreading. So this is also a good way actually to make something useful in Slovenia as well. One important fact: biomass is carbon neutral. It is resorbed and used for growth of new biomass, and this is a fact as well. Typically, cellulose-based biomass is built of about 40 to 50% of cellulose, 20 to 30% of hemicellulose, and 10 to 25% of lignin. Cellulose and hemicellulose is quite simple, but it's, the structure of cellulose and hemicellulose is quite simple actually. It contains some sugars or cellulose, for example, glucopyranose units. That is this stuff here. On the other hand, lignin is quite a complex aromatic polymer, which of course also has some sugars in the molecule, but on the other hand, some aromatics as well. It's a very difficult aromatic polymer. It is difficult to analyze it, and it varies from the one plant to another and so on. But on the other hand, the cellulose is packed in microfibrils. And these microfibrils, as you can see here on this slide, there are some regions which are amorphous, and there are some very ordered regions which are crystalline. And these crystalline regions are actually this famous nanocrystalline cellulose, which we are looking for a very effective production and utilization. Well, worldwide, there are very different ways how to isolate this nanocellulose, nanocrystalline cellulose that I'm talking about. You must know that there also exists a nanofibril cellulose, which is not crystalline and has a very long fibrils. And there is also bacterial nanocellulose, which is grown from the bacteria. But I will concentrate on this nanocrystalline cellulose because this is my expertise and my knowledge. The standard procedure how to do it, how to isolate from, let's say, wood biomass is illustrated on this slide. You can see that you need some steam explosion or milling or something like that, then bleaching, and then your cellulose is subjected to acid, quite strong acid hydrolysis. And this acid hydrolysis is usually made by sulfuric acid with quite high concentration, 65%. And so it means that at the end you have to get rid of this sulfuric acid by dialysis or something like that. And at the end you will have this nanocrystalline cellulose, which also has on the surface some sulfate groups, which is not very popular in some cases. Nevertheless, in Slovenia, we've done some work on this wood liquefaction and so on, and we found out that it is possible to use this wood liquefaction process also for the production of nanocellulose. How does this happen? Actually, we did this wood liquefaction, and we found out that there is some kind of very fine dust clogging our filters. And what was that? Nanocrystalline cellulose. So we actually modified the process to get this very effective production of nanocrystalline cellulose. You can see that it is quite simple process. It consists of milling, cutting to small particles. Then the polymerization takes place with glycols, which are diethylene glycol, glycerol, or something like that. And only 3% of methanesulfonic acid as a catalyst is added. The temperature, you see, it's moderate, and the reaction time is between 60 and 180 degrees. At the end, we have this kind of suspension. Let's say the reaction mixture is a suspension of nanocrystalline cellulose, and everything else is dissolved. So what we need the centrifugation and cleaning with solvents or water or mixture of them. And at the end, we have this nanocrystalline cellulose suspension in water. If the color is, let's say, brown or white brown or something like that, there also need a bleaching with just simple sodium hypochlorite with a low concentration, and at the end you will have almost a transparent nanocrystalline cellulose. On this slide you can see what is how our product looks like. This is the same microscope photograph. You can see very nice little crystallites which are long between 200 and 300 nanometers. The thickness is with around 20 nanometers, so it is really a proper crystalline material. And on the bottom you can see how it is transparent and colorless. And this is the last achievement in our pilot run production. we can produce also the dry nanocrystalline cellulose. If you mix this powder with water, you will get this kind of stuff as it is in the standard way. So how did we do this transfer from the laboratory experiments to pilot plant production? It was actually the first step was the liquefaction of biomass in this 250 liters reactor. It is just the replica of our lab reactor, actually, with mixing, heating, and so on. And it is also possible to speed up the reaction by using high-power ultrasound. This is on this slide. You just put a 4 kilowatt ultrasound sonotrode into the reactor, and you speed up the reaction extremely, actually. You can see that 50% of reaction time goes off and less energy is spent for that. The next step is centrifugation, rinsing with solvent, centrifugation and rinsing with solvent. You can see this continuous centrifuge in this pilot plant production process. This is really powerful centrifuge. You need at least 8,000 or 900 RPM to get proper sedimentation of nanocellulose, because the particles are very small actually and light. So you need a lot of centrifugal power for that. So this continuous centrifuge gives us this. And we also use this ultrasound system for this because you have a sediment and you need to put this sediment again into some kind of dispersion, and ultrasound is useful for that. The next slide will give you some just rough information, actually, what is needed, for example, for one cycle of this nanocrystalline cellulose production. The 3.5 kilograms of the reaction product from the reactor, you dilute it, you centrifuge it, you rinse it several times with mixture of glycol and water or just water, and then at the end bleaching and the final product. It's 10 to 15% suspension of nanocrystalline cellulose in water. So the process is actually quite simple. The yields are quite high because the yields in the lab are about 60%. In the, let's say, pilot plant production, it's around 45%, which is really quite high. And of course, the price for this production is very low. I would say that it is at least 5 to 10 times less than the standard procedure with this high concentration of sulfuric acid. So the process is not the secret, actually. You can use it in your lab, but if you start an industrial production, see you in the courtroom. Some applications, as I promised you, of course, we must. There are several or many literature data what can be done with this nanocrystalline cellulose. When I made some search on the web about application and patents dealing with the nanocellulose, I found that both rise exponentially. So it really is a hot material. The people are looking for biodegradable, biocompatible, bio-based material, biomass additives with some positive result in their process, in their products, actually. And nanocellulose is all of that. I'll show you some applications in the next slides. First of all, nanocrystalline cellulose is a great rheology additive. You can see from this, the first graph, that the viscosity goes quite up by adding just a small amount of nanocrystalline cellulose into the system. That can be some kind of coatings or something like that, or some, I don't know, cosmetic material, some adhesive, and so on and so on. On the other hand, there were already done some very, very successful experiments about this coatings for papers. You can see this on this reference because we use this nanocrystalline cellulose in paper, in coatings for paper, and the result was better mechanical properties and better printability. So again, this is interesting for the paper production. One of very promising area is use of nanocrystalline cellulose in packaging industry. Why? The reason is actually nanocrystalline cellulose is a perfect barrier for anything, for the water vapor, for oxygen, or for grease and oil. Because the point is that let's say, water molecule has to travel through this layer where nanocellulose particles are oriented like this. So it means the pathway is very long. And the reason and the result, of course, is on this graph. You can see how difficult or how air permeability goes down when adding just a little bit of nanocrystalline cellulose in this material. The next applications is in adhesives. You can see that the properties goes up again in polyvinyl alcohol adhesive. Again, the mechanical properties, the wood, the joints are better, and so on and so on. And on the other hand, in this polyurethane nanocrystalline cellulose nanocomposites, again, the tensile strength goes up. I would say that always in all applications, a very small amount of nanocrystalline cellulose is added, 1–2%, not more. Because if you exaggerate with this concentration of nanocrystalline cellulose, you will lose the mechanical properties. It is good actually because nanocrystalline cellulose is not cheap. So if you add just a little bit, it's okay. The next application, which is quite promising, is in biomedicine. They have been studied intensely nowadays since nanocellulose has excellent mechanical properties, is biocompatible, has huge surface area, so it is a good drug carrier actually, and it is not toxic. Membrane carriers can be good for water holding capability, so flexibility is good, and so on and so on. So it means it's good for the, let's say, for binding material, for let's say chronic wounds and so on. On the other hand, what about the, let's say, about the toxicity of nanocrystalline cellulose? Nanocellulose is problematic in dust form. The inhalation of plentiful of dust of nanocellulose may induce some kind of inflammation. But on the other hand, they tested the cytotoxicity on 9 different cell lines and results were always negative. I think that the personal protection gear and avoiding the exposure to dust particles is of course recommended. But on the other hand, if it is in some kind of a gel or something, there is no problem. I must stress that nanocrystalline cellulose has a lot of OH groups on the surface, these hydroxyl groups. It's normal because this is the, let's say, glucose actually, and it has a lot of this. These OH groups are ready to be derivatized in different way. You can see what can be done with these OH groups. Actually, all the reactions that are known that these OH groups go, they can be done on this nanocrystalline cellulose surface. And that means actually that the modification of nanocrystalline cellulose gives you some other advantages which can be then used in, let's say, polymer chemistry, polymer composite chemistry, in I don't know, medicine or whatever. It means that you can modify the surface to be less polar, more polar, have some, let's say, carboxylic groups on the surface and so on. And this can be useful then in different areas. So to finish my presentation, I think that I made quite a, let's say, EPP for this nanocrystalline cellulose. I must say that we have this plant production in Slovenia. I think that nowadays the production can be also already about 200 kilograms of suspension per day. It is, let's say, quite cheap regarding the other producers in the worldwide. And of course, the properties of this nanocrystalline cellulose, I think they are really unique. And at the end, I can say that the future about these bio-based materials, bio growth, actually, I think it depends only on good fertilizer. Thank you for your attention.
Thank you very much, Matjaž, for your very interesting presentation. And our third and last session is going to be presented by Valantis Ketikidis from Serth, Greece, who will speak about optimal use of waste streams for biogas production. So let me switch the microphone to you, Valantis, and would you please continue in a few seconds? There you go.
Thank you, Natalija. Hello, everybody. My name is Valantis Ketikidis, and I am from the Center for Research and Technology in Greece. The center is based in Ptolemais in Western Macedonia, and the headquarters are in Thessaloniki. So why do I mention this? Because Western Macedonia is the region which is participating in the Bioeconomy project, which is a territorial cooperation project. So my presentation today is about the optimal use of waste streams in the biogas production. First of all, let us see the Venn diagram of the bioeconomy. You see here the green economy. Within this green economy is the bioeconomy, the bio-based economy, and the circular economy. There's a cross-cutting and overlapping area between the bioeconomy and the circular economy, and somewhere there is the biogas production. So we have the contribution of this biogas to the bioeconomy / circular economy. Why do we mention this? Because the biogas is improving the sustainability of industries by extracting the energy from their own effluents and using it for the self-generation either of electricity or electricity and heat. Of course, it is also recirculating the nutrients and the organic matter in the organic wastes, which are utilized for the anaerobic digestion process, and returning them to the soil in the form of biofertilizer. You see here the anaerobic digestion process. This flow chart is showing how we reach the production of biogas. We start with the organic waste, either from the farm residues, food residues of the sewage. Going through the anaerobic digestion process, we produce biogas, which will be further utilized either in the CHP unit to produce electricity and heat, or it is upgraded further on as biomethane and injected into the gas grid. As a byproduct, we have the digestate, which is further utilized as a solid, as a fertilizer for the crop production. So you see here, we have a very good cycle, which is closed. Some words about the biogas utilization in Greece. So despite the huge raw material we have in this country, the Greek biogas sector is still in its infancy. So this is due also to the Greek economic crisis of the last years. We had financing investments which have been suffered. However, there are there's a considerable number of biogas plants all over the country, which are 33 almost in 2018. Regarding the total installed capacity, almost 14,000 kilowatt electric are coming from plants at municipal wastewater treatment facilities. Another 22,000 kilowatts are coming from agricultural wastes, and 25,000 kilowatts are coming from municipal waste landfills. So you see here the majority of these plants are belonging on a municipal level. These Greek biogas plants, they run usually on animal excrement, slaughterhouse wastes, or fermentable waste from agriculture or the food industry. And as said, Western Macedonia is the regional authority, or the region, which is the geographical region, which is participating in the Bioeconomy. You see here in the circle, we have 4 biogas production facilities as of 2020, and around 1,500 kilowatts from agricultural wastes. Next slide is about an overview, to have a small picture about what is happening all over Europe. You see here the number of biogas plants in Europe as of 2015. Greece, in contrast to the other countries like Germany or Italy, it's not only a differentiation of the number of plants built or in operation, it's also a differentiation of the feedstock used. You see here, Germany and Italy have a lot of plants, a lot of more plants. Also, they do run on an agricultural basis and not that much of landfill or sewage sludge, which is on a municipal level. So you see here a very nice balanced development on a regional agricultural level throughout the country, as referring to Germany and Italy. Next slide is the estimation of the biogas production from agricultural residues and livestock manure. You see here, as said, the feedstock used is coming from the agriculture, from the residues from the agriculture, like cereals, like citrus fruits, olive oil industry residues, dairy industry, and concluding to the livestock manure. The biogas yield is varying from 0.4 cubic meters per kilogram volatile solids to almost 1 when referring to the dairy industry. Of course, you see also here, when referring to the livestock, a high amount of biogas yield. However, you have to take into consideration that the harvestability of the manure is not considered. So you see here the theoretical potential of biogas and methane production without considering the harvestability or the feasible technical potential, which is lower than this here shown. Some barriers regarding the biogas growth. So the main barrier for the growth of biogas here in Greece is the development, is the support scheme in the heating sector, which is almost inexistent in contrast to the support scheme, the incentives which are provided for the electricity sector. So this is the main barrier. However, we do have also a lack of efficient and reliable supply chain. The most feedstock which is used for the biogas production is located in numerous places. The morphology of the country, with the many mountainous areas, the fragmented location, and the small size of the farms, mainly animal farms, this does restrict the identification and the location of the feedstock sources. Then we have a lack of reliable information sources. There are almost 100 secretariats in charge of collecting data of these available feedstock, and often the information remains fragmented, even if we are talking on a similar organic residue stream level. There is a lack of expertise, especially when we are talking about the personnel in authorities who do not have the necessary technical background as to evaluate the quality of the information provided. And the same goes also for the personnel of the administrative regions, which is responsible for the authorization of the installations. However, there are also some key drivers of the biogas development which are related to the policy amendments. So we have a subsidy system here in Greece, and this is favoring the generation and injection of electricity from biogas into the grid, because of the establishment of a sustainable industry, because it's contributing to solve the existing waste disposal problems, it's making use of the existing raw material, and contributing, of course, to the climate protection. There is a premium tariff established in 2016, the feed-in premium, It is referring to the RES and CHP plants to be connected to the transmission system, and which is participating in the electricity market and is awarded a sliding feed-in premium. From 2017, this feed-in premium is granted through tenders. So the support contracts for wind power, for example, of plants which are above 6 megawatts, or other RES plants where the biogas is belonging above 1 megawatt, these are on hold until a tender is announced. Of course, there are also exceptions to apply to smaller installations where we have an eligible feed-in tariff. So this feed-in tariff, it is a 20 years state fixed feed-in tariff. And it is considering the biogas produced according to the installed capacity and of the power plant and its substrate to be used. So you have here landfill biogas, for example. It's coming from fermentable waste and organic sewage sludge from wastewater treatment. See here we have another feed-in tariff, then the biogas from the biomass originating from organic residues and wastes from livestock and agriculture, which has a higher feed-in tariff. So these tariffs only apply to new installed electrical connections up to 50 megawatts per year. Once this number is reached in a calendar year, tariffs for electricity from biogas are tendered until the end of that year. So this process is in effect from 2017, as already mentioned. And now we are going back, or we are going further to the new investment law, another policy amendment, which is a driver for the biogas development. It is also in effect from June 2016, and it foresees a support for the development of biogas in the form of income tax reliefs, subsidized expenditures, leasing subsidies, etc. And it is referring both to electricity and heat generated. So back to the technical description of the anaerobic digestion process. You see here a very nice flow chart. The anaerobic digestion is a complex process in which organic material is broken down by several groups of microorganisms in the absence of oxygen and under certain conditions. So we have conditions regarding pH, total solids, volatile fatty acids, total Kendall, nitrogen, ammonium, alkali, and the COD, the chemical oxygen demand. So the biogas generated from the AD process is renewable energy. This is what we have discussed just now, consisting of methane and carbon dioxide. And as regards this complex microbiological process, we have 4 stages. We start with the hydrolysis, going further to the acidogenesis, the acetogenesis, and concluding with the methanogenesis, where we have methane and CO2 carbon dioxide. There are 2 optimum temperature ranges, the mesophilic from 35 till 40 degrees Celsius, and the thermophilic, which is around 55 and 60 degrees Celsius. There are some significant differences between these 2 operating modes. Most AD plants around the world, they operate on the mesophilic range, as less heat is required to maintain the temperature, and also the digestion process is more stable under these conditions. The thermophilic reactors are sometimes installed as they accelerate the degradation rates. They create higher yields of biogas and reduce the pathogens in the digestate produced. Of course, they have a higher operation running cost. Now to the optimum design and the efficient operation of these biogas plants. You see here we have a diagram regarding the classification of anaerobic digestion technologies. And this classification is based on the constituents and the consistency of the waste treated. So we have anaerobic digestion, which are referring and designed to wet, dry, or liquid digestion systems. So on the left side, you see here the digesters which use as a raw material or as substrate, as we define it, the wastewater, which has a low solids content around 4 and 10%. And on the other side, you have here the treatment of slurries like manure, like dairy waste, organic solid waste, which has a higher solid content. So on the one side, we have the wet digestion, if the solid contents are below 20%, and the dry digestion, if the solid contents are higher. And you see here the hydraulic retention time is also differentiated according to the consistency of the waste treated. So we could say that the pretreatment needed, the digesture configurations and operating temperature, as well as the need for pasteurization, they vary based on type and quality of the feedstock, of course, on the climate and local economy, because they differentiate the raw material to be used. So each digester is designed and optimized individually. So the role of the inoculum, what is the inoculum? As we said, microbiological process is going on in digester in the biogas plant. So bacteria are very sensitive to environmental conditions. It is important to balance a range of factors to maximize the chances for achieving optimum design and efficient operation. It's important to know the characteristics of this organic material to be used, to be digested, and often this is rate limiting. It can be inhibit the whole process. So the feed characteristics such as toxicity and biodegradability are major factors for affecting system design and performance. So how do we proceed, or how do we accomplish this optimization of the biogas production facility or biogas production process? We use the biochemical methane potential tests, the BMP tests. These determine the methane potential and the biodegradability of wastewater and waste. So the investigated material is mixed with active anaerobic inoculum collected from a full-scale digester. The mixture is incubated either in mesophilic or thermophilic conditions and well mixed for optimal mass and heat transfer. You see it here, the BMP test. The test continues to run until the material is considered fully degraded or the daily gas production is less than 1% of the accumulated gas production, and this needs around 30 until 60 days. The whole process is analogous, is dependent on the physical and chemical properties of the material and the activity of this inoculum. So during the test, the volume of the produced gas is measured. And again, back to this device, you see here a schematic diagram of automatic methane potential test system. This is this device here. It has a lot of spaces, a lot of places in order to accept various samples with a lot of diverse substrates and various operational conditions. So this methane potential test system, it is measuring the volume of the gas produced and is based on the principle of liquid displacement and buoyancy. You see here the diagram. The desired component is the methane. CO2 is retained here, is still remaining here. At the end of the process, the BMP test, the biochemical methane potential is calculated and adjusted to standard temperature and pressure under dry conditions, and there is the accumulated methane potential curves are plotted. So the BMP tests, they have been used to characterize a wide variety of substrates, either liquid or solid. As we said, depending on the total solids content, we have various technological standards of the digestion process. They have been important tools for investigating possible pre- and post-digestion treatment options. These batch experiments, they have been found to produce reasonable predictions of a full-scale behavior. And the BMP tests of the substrates to be digested and their specific organic loads could be used to design different components of a full-scale anaerobic digestion plant, such as the size of the digester and the possibilities of exploiting the produced biogas. The whole procedure is based on some standards. We have many international and national procedures which have been proposed, each one of them using different serum bottles, test inoculums, inocula, food to microorganism ratios, nutrients, methane measurement devices. The most popular methods are from Müller, from Hansen, Angelidaki, and the Association of the German Engineers Standard Procedure, the VDI. The conclusion. So a small recap of what we did say until now. The BMP tests, they provide insight into the key factors such as the inoculum, the substrate, experimental conditions, operational conditions, and data analysis reporting on anaerobic biodegradability in batch mode. More detailed, they conduct toxicity assays and determine the true biochemical methane potential and the dynamic degradation profile of the biomass substrate. They allow users to more easily determine the optimal retention time and the mix of these substrates to be co-digested. They screen proper pretreatment methods, very important, and they evaluate the need for additives, if so. And this data can be used to extract important kinetic information of the degradation process, which in turn provides for a much better understanding of the dynamic degradation behavior of a specific substrate. So this slide concludes my presentation. I thank you for the attention, first of all, and I give the floor back to Natalija in order to coordinate the rest of this webinar. Natalija, please, you have the floor.
Thank you very much, Valantis, for your very interesting presentation. And with Mr. Ketikidis' session, we finalized today's webinar. As you have seen, we are working on bioeconomy applications of bio-based products, be it aquatic or wood biomass or biogas production. I thank you all for attending. We hope you found it useful, and we would like to hear from you in the future, so please do not hesitate to contact us on any of the topics presented. I hope you have noted our emails at the beginning of our presentations. Also, we have recorded this event, and you will be able to download it from your BBB homepage in a few minutes. So thanks again, and have a great rest of the day. Bye-bye.